So it's 9:01 a.m. on the East Coast here in the US. So I think we'll get started. We have quite a few attendees already, so I think we're good to go. So I wanted to start off by saying hello to everyone. Welcome to Salvivo's seminar series. Thank you very much to everyone for joining us for our second webinar from all over the world. For the second installment, we have Professor Doris Wilflingseder from the Institute of Hygiene and Medical Microbiology at the Medical University of Innsbruck. She will talk to us about the use of complex and predictive non-animal models to gain more specific insights on host-pathogen interactions and potential treatment strategies. Before I get started introducing our speaker, I wanted to ask all participants if you have any questions as Doris goes through her material to please submit them into the chat function, and we will address them at the very end of the talk. So to get started, I'll provide a brief introduction for Doris. Doris's research group at the Medical University of Innsbruck is interested in modulation of dendritic cells and macrophage function by the opsonization pattern of pathogens in relevant 3D cell culture microenvironments. To address these questions, they use molecular biology tools, imaging and immunological approaches combined with primary cell models, as well as some appropriate cell lines. So please welcome Doris, and I hand it over to you.
Okay, thanks, Riham, for this kind introduction. I'm really looking forward to showing you some of our results, and I'm happy to welcome you all to this webinar. So as Riham has said, I'm interested in host-pathogen interactions, and together with our groups, the Associate Professor Wilfried Bosch and I, so here you can see our groups, we are interested in various human pathogens. So, for example, we are interested in HIV-1 and HIV-2. We are interested in coronaviruses or other respiratory viruses, but we are also interested in co-infection models with respect to viral fungal or viral bacterial co-infection. And since these are all human pathogens, we were thinking to really optimize and standardize human barrier models of mucosa, lung, and skin, and equip those with components of the immune system which are relevant at barrier sites. For example, dendritic cells, macrophages. Recently, we also introduced neutrophils in our models, but we are also interested in components of the humoral innate immunity, such as the complement system. And in addition, we can also add T or B cells to our systems, which I want to introduce to you within the next minutes. And these models in our lab, we established and optimized. And now we are offering a standardized platform of host pathogen interaction. And this we combine also with data from patient materials, because we have a very close collaboration with the internal medicine too at the Tirol Kliniken here in Innsbruck. And therefore we, in particular during COVID-19 times, we got really nice materials from either convalescent patients or also vaccinated patients to also test saliva and serum from such individuals within our systems. So therefore, we also have a really nice translational collaboration. And I don't want to go into the COVID-19 infection process too much into detail, because we've heard quite a lot over the last 3 years, but I just want to remind you that common cold coronaviruses, such as those strains shown here, and also the Omicron variants, which we now are facing since about beginning of this year, are more likely affecting the upper respiratory tract, while those viruses which are affected with a more severe pathogenicity, such as SARS-CoV-1, MERS-CoV, or the earlier variants of SARS-CoV-2, more likely affect the lower respiratory tract. And that's why in our lab, we established models throughout the whole respiratory tract down to the lungs. So we established models of nasal origin, of upper respiratory tract origin, a small airway epithelial model reflecting the lower respiratory tract. And we also have a lung model, which I will show to you in a minute. And this we don't only have from healthy individuals, but also from diseased states, such as cystic fibrosis or COPD. And that's why we can compare infection processes within healthy individuals versus also diseased individuals. And I want to show you our models right now. And this is our air-liquid interface models, where we use primary epithelial cells. So we do not use cell lines because these often cause unspecific migration of immune cells when immune cells are applied to the model. So therefore, we are using primary epithelial cells of primary origin, which we first expand until about passage 3 in T75 flasks, because this is the passage where we found out that those cells which are of primary origin and which are not immortalized still have the same features as from the first passage. So therefore, we first expand the primary cells within monolayers, and then we freeze most of the cells. The other ones we are culturing in the next step on transfer filters that we are coating with a birch-based cellulose, because we wanted to get rid of really all animal components from this system. And usually or often red tail collagen is used as a scaffold, but we also wanted to get rid of this. And so we compared the growth and differentiation of those primary epithelial cells directly in parallel in red tail collagen versus the birch-based cellulose, and really characterized the proliferation as well as the differentiation of such cultured cells really in detail to get standardized conditions. And in the end, we found that within the birch-based cellulose, the cells differentiated faster, so we could use them faster for our experiments. So therefore, we did not only omit the animal component in our system, but we also have an accelerated differentiation of the cells within these cellulose. And another advantage of the cellulose is that you can easily use cellulase to digest the cellulose and make single cell analysis again. And then we culture such differentiated cells in air-liquid interface. So here we have the air phase, here we have the liquid phase, and these are fully differentiated epithelial cells of respiratory origin, which contain basal cells, goblet or club cells, depending on where from the respiratory tract they are, and also, of course, ciliated cells. And within this system, it's quite easy to introduce the immune component. So we tested the system with dendritic cells, with macrophages, with neutrophils, and here you could also add T or B cells to the basolateral side. So therefore, you can see that this system is really quite physiologic because it's in an air-liquid interface, and it's really easy to also add immune cells. Of course, you can also add humoral components such as complement. So from this system, we can get from one filter really lots of readout parameters because on the one hand, we can measure the transepithelial electrical resistance to see on whether the epithelium is intact and whether its integrity is good, because the higher the resistance, the better the epithelium is feeling. And whenever you infect it, for example, with SARS-CoV-2, the epithelium gets destroyed, you get holes, and therefore the tear value is strong. Then we do lots of live cell imaging analysis, and we can also do live cell imaging to look at the mucociliary clearance. And from the basolateral subnatant as well as from the apical side where we have the mucus, we can perform ELISAs, we can measure pro-inflammatory cytokines, we can do detection of viral loads, which are either released to the apical side or to the basolateral side. We can do the testing of the infectiousness of the viral particles released by doing plaque assays. So you can see that we have quite a lot of readout parameters just from one filter. But of course, such a system is not really appropriate for high content screening of lots of drugs or possible antiviral sprays. So, but this system nevertheless can be used for testing in a patient-specific manner, because when we have this fully differentiated system, which you can see here, it takes about 3 weeks that we have such a air-liquid interface models. Then we can treat these systems with saliva or serum from patients, which we, as in the beginning I said, which we get from the clinics. And then we can add the different variants of concern of SARS-CoV-2, for example, and test on whether, for example, the vaccination is effective against also different variants of concern, against newer variants of concern, on whether we have salivary antibodies which are effective against the variants of concern, or we can do quite a lot of tests within this system. And this is how the system looks like when we do immunofluorescence or microscopic analysis. Here you can see the ciliated cells in green and the mucus and mucus-producing cells in orange. And you can see here a blurry matter, and this is the mucus produced by the cells. And here on the right-hand side, you can see a nice live cell analysis of the ciliated cells. So this system, the other system we are using are lung organoids, which are normally growing with the basal side out in the medium, and they have the apical side in the center, as you can see here, because here we screened for the SARS-CoV-2 receptors ACE2 and TMPRSS2, and you can see that they are in the center. Here in green, the TMPRSS2, in pink, the ACE2, in orange, we also stained tight junctions by using SO1, and the nuclease were stained in blue. During the first wave of COVID, we found a protocol, a published protocol, which described to reverse the polarity of such organoids into an epicloud phenotype. And this is what we then tried. And this protocol really worked nicely, so that we now have the apical side out, as you can see here from the cilia beating, and the basal side in. And this is also the more physiologic manner to infect these lung organoids. So therefore, we have those two complementary systems. On the one hand, the more physiologic system of the air-liquid interface, where we can easily introduce immune cells. On the other hand, the organoid system, which we turn into an epicloud polarity, and which is more appropriate for high throughput. And also this system, we wanted to get rid of really all animal components. And as you know, organoids are mostly cultured in gel tracks or matrigel, which are derived from animal origin. So therefore, in this case, we did a direct comparison of the growth and differentiation of such organoids, of apical out organoids within gel tracks. So we use the gel tracks or matrigel versus the growth within the cell vivo, within the clinostar of cell vivo, which has, there you don't need any scaffold. You have those bioreactors which are moving within the incubator, and you can also turn the movement of those bioreactors. So therefore the cells are always in movement. So therefore they easily self-organize. And we directly, as said, compared those two methods because we wanted to get rid of these animal-derived components, which are also of an undefined composition and which we really wanted to also get rid of from our system. And therefore we did this, we always do those direct comparisons to the gold standard, which is used in the cell culture. And what we saw here, so on the one hand, we looked at basal out cells and at apical out cells, which were on the one hand cultured in gel tracks, on the other in cell vivo. And we could see that the cell vivo, the clinostar, was superior in differentiation of ciliated cells. It was also similar and not significantly different in terms of basal cells. And also we could see really within the cell vivo, we had a really nice differentiation of those cells. This is now just a very rough overview, but we did in recent time more characteristics and more comparisons. And what we found was that in particular within the proximally differentiated organoids, they were really growing nicely. You see a faster growth, and we could see much more ciliated cells, and also mucus producing cells were expressed in such organoids. And therefore we are in particular for the experiments which we are doing constantly, and where we always need some aliquots of organoids, we prefer to have the organoids which were cultured within the cell vivo without any additional matrix. And therefore we now did this more detailed characterization. And we think that we now also optimized these conditions, and with those two systems, which, as I told you before, really complement each other nicely, because on the one hand we can do high throughput with the organoids, on the other we can do more physiologic conditions. We asked several questions over the last years, in particular in the first phase of COVID-19, where we got the first isolates of COVID-19 patients which were in the clinics. We wondered in the very beginning, where nothing was known on what's really happening at the barrier sites, what happens really right after SARS-CoV-2 entry in respiratory tissues. And what we found there was on the left-hand side, you can see a non-infected tissue. On the right-hand side, you can see a SARS-CoV-2 infected tissue. We could see that the SARS-CoV-2 infected primary human airway epithelia illustrated an extensive tissue destruction, as can be seen here. We also measured, of course, TARE values, which were significantly lower in such infected tissues versus the uninfected. And what we also found, and this was really interesting, was that we saw a mucus hypersecretion, which you can see here in orange. Here you can see really a large areas of mucus secretion, and also mucus plug formation, beside, of course, infected cells, virus particles, and, as said, destruction of the epithelia. While here you can see the normal mucus production, you can see some mucus producing cells, but you never see this mucus hypersecretion. And sorry, just to tell, this mucus hypersecretion was also described in critically ill COVID-19 patients at that time. So therefore, we thought that this relatively, this relatively not so complex system, which just exists of a differentiated epithelium, nicely reflects what's happening at barrier sites also in COVID-19 patients. In newer times, we wanted to look at the differences which are occurring when, for example, we are infecting a tissue with Delta versus Omicron. And the virus you can see here in pink, and we also stained for an innate immune activation marker, which is complement C3. And what we could see was that Delta penetrated really to the basolateral side of the pseudostratified epithelium, which can be also nicely seen here in this ZY axis. So we saw really a deep penetration of virus particles to the basolateral side upon Delta infection, while we found that Omicron infection really happened superficially at the apical side of those tissues. And we could also see that only upper layers were destroyed by Omicron, while Delta really caused a massive tissue destruction in those cultures. And we then also measured the distance of viral particles from the apical side, and this was significantly higher in Delta versus all the Omicron variants we tested. And this is in the meantime also published. And we also found that the original SARS-CoV-2 variants, such as the wild type variant in the beginning, Delta, they really also caused a very high innate immune activation reflected by complement, which was released to the basolateral side of the epithelium. And this also, of course, then also causes migration of immune cells to the sites of infection and causes harming of the tissues. So therefore, we wanted, this was also seen in severe COVID cases in the beginning of infection. And we also described this and also published this already in 21. And this was also something which we wanted to see whether we can somehow stop this high cytokine secretion or high innate immune activation at barrier sites already. And this is what I already showed you, that C3 is this complement C3 is highly induced in the air liquid interface system, but also in the lung organoids. And this was also accompanied by a drop in the epithelial resistance and, as said, a release of the anaphylatoxin C3A. And this was significantly higher in SARS-CoV-2 infected tissues. And therefore, we asked on whether we could stop this reaction, whether we could stop this innate immune activation by adding complement receptor inhibitors. And this is what you can see here. So here, this is the best example. The C5a receptor inhibitor worked best. You can see that we nearly have no inflammatory response in terms of C3 in the tissues anymore. The tissues look really similar to uninfected tissues. And surprisingly, also with the omission of the inflammatory response, also the viral load really significantly dropped, which can be seen here, because here we just have the infected tissues where the virus is shown in red, and here we have blocked C5a receptor at the basolateral site of those tissues, and this also resulted in a significant drop of the viral load. And this is what we also determined using quantitative analysis by determining the viral load. So we also had a significantly lower viral load in such samples treated with C5a receptor inhibitor. And we also had a significantly reduced anaphylotoxin secretion. And this block in the very first innate immune responses within epithelial cells was also associated with a significant down modulation of other pro-inflammatory cytokines, which were associated to having an impact, of course, on the cytokine storm seen in severe COVID-19 cases. So therefore, we proposed that inhibiting the complement at the beginning could also be a good means to down modulate the cytokine storm. And this was also done in the very first COVID-19 wave with patients in Italy, because at that time no corticosteroids were given, so therefore we did not have really any clue how to stop this. And therefore in Italy there were case reports where they added complement inhibitors, and by this could really rescue the patient's life. So therefore we think that the system nicely also reflects also reflects reactions going on in the patients. And when also always comparing it with patient data from the literature, this also makes the picture rounder. Also, this is, as said, not such a complex system, because here are no immune cells added. We also, as I showed you before, had a cohort study running on serum and saliva neutralization, and this would also result in a personalized protection assay, because then we can determine on whether, for example, the vaccination was efficient and on whether the vaccination is also efficient against newer variants of concern. And as I showed you before, we use serum and saliva from individuals which were either 3 times vaccinated and then were infected with Omicron, at that time Omicron BA4 or 5, or we had a 3 times vaccinated group and who got a bivalent booster. And from such patients, we had the serum and saliva 5 to 7 weeks after the last immunization. And we were interested on whether we see neutralization of SARS-CoV-2 variants of concern from the saliva or from the serum, and added either saliva or serum, which had on high IgGs or IgAs in case of saliva, and low IgGs or low IgGs and IgAs in saliva. And what we could show was that that saliva samples from the high titer group, which are shown here in dark blue, diminished viral titers in all individuals to 0 and maintained also the tissue integrity, which is shown here. And also here the tissue integrity, it was not as high as with uninfected samples, but nevertheless significantly higher as compared to infected samples, or also compared to samples where saliva samples showing low antibody titers were added to the apical site. And what we also found was that addition of high titer serum resulted in partially reduced viral particles and also increased tissue integrity. So therefore, we can say that this system can also be used for making such personalized assays. We also, in addition to those analyses before, did immunofluorescence analysis and found a highest reduction of virus and complement signal in the high titer saliva samples, which coincided well with the other tests in our system. And also the addition of high titer serum reduced the viral signal. So therefore, we think that this system is also really nice to test in a personalized manner the capacity of IgAs or IgGs in terms of their neutralizing capacity. And therefore, I think this is a quite nice physiologic model to have a good result on the neutralization of such samples. And with this, I'm already at the end of my talk, and I'm really happy to take your questions. And just as I thought I would need even longer for this presentation, but I just wanted to tell that we, as I told you in the beginning, we also have now new analysis where we added immune cells to the system, and where we also can look now on what's happening on the immune side, and that's what we are working on right now. So thanks for your attention, and I'm happy to take your questions.
Doris, thank you so much for your talk. I want to encourage all participants, if you have any questions, to please submit them. But in the meantime, I think we have a few that we can get started with. The first question, Doris, is, can you tell us the specific protocol for how to generate apical out organoids using the Clinostar?
Yeah, what we are doing is that we are, so at that time we in the Clinostar.
Mm-hmm.
In the Clinostar.
Yes.
Okay. The exact protocol. I have to say I have to check. We are changing the medium composition in the Clinostar to get an apical out phenotype. I can't tell the exact composition right now, but we leave the organoids within this medium for 1 to 2 weeks, and then we get this proximally differentiated phenotype in the Clinostar. And if we want to publish this anyways pretty soon, so we now did several more characterizations in terms of the differentiation of those cells, because we did not only check the markers which I've shown here, but checked for several more markers, and we also checked for another culture because we also used, for example, an animal-free scaffold where we had the differentiation. So we did a comparison of the gel tracks versus an animal-free component versus the Clinostar, and we are right now writing that up, and this will be published soon. And if the protocol is needed, of course, you can also drop me an email.
Great. Another question just came in, and it starts off by saying captivating microscopic images, which I have to agree, Doris, your images are absolutely breathtaking.
Thanks.
I'm curious to know if your research has explored potential alterations in cilia beating patterns post-SARS-CoV-2 infection.
Yes, so we could not do live cell analysis there, but what we found was that on the one hand, we just from microscopically checking this, we saw a different cilia beating, and what we in particular saw was really this high mucus secretion. So this came hand in hand, that you really saw differences in the cilia beating as well as in the mucus secretion, because in the samples infected with SARS-CoV-2, we really, for example, when we took the supernatant from the pical site just to make viral load analysis or a cytokine analysis, you really saw those plugs. And this is what we also then saw in the immunofluorescence image. We have this mucus hypersecretion and plug formation. And what was also interesting was that, for example, in terms of cilia and cilia beading, because we also treated those filters apically with a antiviral sprays, which we can buy commercially in the pharmacy. And when we treated those with the antiviral sprays, we really saw a significant acceleration of the cilia beating within the system by treating them, which in my opinion is somehow reflecting that maybe the cilia then beat faster and get the virus out faster from the system, or somehow by this faster beating somehow hinder the attachment of the viral particles to the target cells. And this was really interesting, because these analysis we could do in a live cell modus, because we just added fluorescently labeled beads and we treated the cells with the antiviral sprays or with a control component, then added fluorescently labeled beads, and then looked at mucociliary clearance, and there the speed of the cilia was really significantly higher than in the control wells.
Thank you, Doris. Another question, what kind of 3D cell culture model did you use for SARS-CoV-2 study?
We used both models. So on the one hand, we used the lung organoids and in particular the apically out polarized organoids. But on the other hand, we also used the air liquid interface model. So we always checked it within both models. We then also used models of different origins, so of nasal origin, of upper respiratory tract origin, of lower respiratory tract origin. So we tested in particular in the beginning, where we did not know quite much about the whole interaction of SARS-CoV-2 with the respiratory epithelium. We used all the different models, so from nose down to the lung.
Okay, I think those were all the questions we had for you today, Doris. Thank you so much for your talk and your absolutely gorgeous images. Thank you.
Thanks.
We learned a lot, I think, from your talk. And to all the participants, thank you so much for joining us today. I also wanted to invite you on December 6th at 10 a.m. Eastern Standard Time for a talk and presentation by Dr. Simone Sidoli. So I hope you will join us on that day for the talk, and thank you again.
Thanks.